Hello dear students, welcome back to PharmWise Academy. In our previous lecture we have studied about the cell membrane, the structure of cell, etc. Now we are moving ahead in the same topic. We are continuing with the part 5. Where in this topic we will study about the nucleus and endoplasmic reticulum, etc. and Golgi apparatus. Okay, we can also include Golgi apparatus. So let's get started. So students, in our previous lecture I have already discussed about the structure of the cell, function of the cell and the structure of cell membrane and what it contains. So now let us begin with the nucleus. We all know the basic thing, the nucleus. The nucleus is present at the, where it is present? The nucleus is present at the center of the cell. Suppose this is cell wall, then this is the nucleus, and this is the figure, the 3D diagram what I am showing to you. Now anatomy, structure. Okay, let us understand the structure. It is called the nuclear envelope, the outer covering, which is can you see the whole this circular thing? This is the nuclear. This is called as the nuclear envelope, and in this nuclear envelope there are the tiny holes, micro holes or poles are present, which is called as the nuclear pores. Okay, by this small nuclear pores, these small substances can easily pass through, but the larger substances such as RNA cannot pass. Okay, the RNA molecules cannot pass through the nuclear pore, but the smaller molecules can pass through. Okay, now inside the nuclear pores, at the center of the nuclear membrane or nuclear envelope, there is the presence of nucleolus. And what is the nucleolus? Inside the nucleolus there contains the chromatids. Okay, chromatids or chromatin fiber. Do you understand the chromatids, or what do you mean by chromatin fibers? Okay, the chromatin fibers are nothing but the DNA strands. Okay, DNA strands, the long DNA strands when are attached, that forms the chromatin fibers. Inside the chromatin fibers the DNA strands are present. Okay, now DNA contains the genetic information. Okay, for all of the living organism the genetic information is needed for life processes. Okay, in humans we have DNA. In some microorganisms there might be present RNA. Now without going to deep discussion in that, let us understand the nucleus, about the nucleus. Okay, inside the nucleus nucleolus is present, and inside the nucleolus chromatin fibers are present, and in the chromatin fibers the DNA are present. Now when, for example, why the DNA is needed? It is a genetic information. It stores the genetic information. For example, if a particular function has to be happened inside the cell, for that particular function to be performed, a chain of amino acids, which we often called protein, a protein molecule is synthesized inside the cell to perform a particular function. Okay, there is a mechanism of performing a function. Okay, and that mechanism involves the synthesis of protein. How that will be work, I will be just demonstrating to you. For example, if a particular message A has to be transferred, like for example, let us take hypoxia. Okay, do you mean by hypoxia? Shortage of, where oxygen concentration in our blood decreases, that we call as the hypoxia condition. In the hypoxia condition, a message has to be sent to the cell, that what the message has to be sent, that the oxygen concentration is getting low. We have to store more, we have to take more glucose, right? Now a particular message has to be sent. That particular, let us assume that particular message sent is supposed to be a message A. Now from where the message is generating means from the DNA. The DNA molecule contain a particular base pairs. Okay, you know, adenine, guanine, thiamine, uracil, etc., etc. That particular base pairs, a particular DNA strand, a particular DNA fragment is responsible for that. A particular gene will be there now, and when that gene expresses, the particular result will be getting. For example, now let's suppose say a message A has to be generated. Now first, the DNA contains, as our humans have DNA as the genetic material. Now first, the DNA molecule, okay, the DNA molecule is a double stranded molecule. Okay, the DNA molecule, it will first get converted to the messenger RNA. Okay, messenger RNA, mRNA. How it will be converted? By the process of transcription. With the help of the process of transcription, the DNA is converted into the messenger RNA. And when this messenger RNA is converted, the messenger RNA is synthesized inside the nucleus. Okay, inside the nucleus only, the DNA is converted into the messenger RNA. The transcription involves 3 steps. However, you will be going to study this in the biochemistry in your upcoming classes. For timing, it contains initiation, chain elongation, and termination. A particular strand of DNA will be converted to mRNA, and from the mRNA will travel across the nuclear membrane, and it goes to the ribosome. Okay, what do you know ribosomes? Ribosomes are the minute structures which are responsible for the synthesis of proteins inside the cell. Okay, so ribosomes will be synthesizing proteins. How? The what sequence of amino acids should be connected? There are 20 essential amino acids. Okay, 20 essential amino acids are there. In what sequence that 20 amino acids should be arranged to synthesize a particular protein molecule? That will be decided by the mRNA. Okay, now this mRNA will go to the ribosome, and this mRNA will be converted into the protein molecule. Okay, this mRNA is going to be converted into protein molecule by the process of translation. Okay, by the help of translation, the mRNA is going to synthesize a protein. For example, if a protein molecule contains, there will be a lot. I am taking just 3 example. Okay, a 3 amino acid, let us say 1 2 3 amino acid pairs have to be arranged, and they should be bonded with each other. Let us suppose in a cell molecule, a nucleus, mRNA is synthesized, it is get outside, it is giving message to, suppose this is ribosome. Amino acid 1 is present here, amino acid 2 is present here, and amino acid 3 is present here. At the site of ribosome, at the site of protein synthesis, these all the amino acids has to be arrived to this site of synthesis, and they have to be arranged in a particular sequence. So they are dispersed very far from the ribosome. How they can be brought near to the site of at the site of synthesis? Well, that is performed by the tRNA, that is transport RNA. The transport RNA molecule will bind to a particular protein, a particular amino acid, that will transport, that will bring and give it to ribosome. Again, sequentially, the tRNA will bring all the amino acids present inside the cell, and a protein molecule is synthesized, and this protein molecule gives a particular function by altering some of the functions, by altering some there are certain mechanisms are there, and thus a gene expresses itself and a particular function, and this function is performed. Okay, did you understood, students? A simple thing is there. Now, basic things often located at the center of the cell, and a important point, don't miss this: mature RBCs do not have any nucleus. Okay, did you followed? The mature erythrocytes, do not contain any nucleus. You will study why it does not contain, because in your second unit we are going to study about the formation of erythrocytes, formation of red blood cells, WBCs, etc. There you will study in deep why the nucleus is not present in the mature RBCs. It contains genetic material such as DNA or RNA. Okay, right? So can we move towards next slide? Okay, we'll move to the next slide. The next is the endoplasmic reticulum. Okay, this is a another organelle present in the cell membrane. What you are seeing, this is the nuclear envelope. Okay, this is the nuclear envelope. The endoplasmic reticulum is a network of flattened tubules. Okay, can you see here? These are the flattened. Okay, these are the flattened tubules which are extended from the nuclear membrane, and it is connected to the cytoplasm. Now, the endoplasmic reticulums are of two types. If on the surface of the endoplasmic these tubules, if the presence of the ribosomes is there, then that endoplasmic reticulum is called as rough endoplasmic reticulum. And the flattened tubules of the endoplasmic reticulum which does not contain any ribosomes, then that is called as the smooth endoplasmic reticulum. So what is the basic difference? Why the presence or absence of the ribosomes will create such a huge difference? We will study now. First, we can take the rough endoplasmic reticulum. Okay, rough endoplasmic reticulum. On the flattened tubules of which are extended from the nuclear membrane attached to the cytosol, which contains the covering of ribosomes, then those endoplasmic reticulum are called as rough endoplasmic reticulums. The main function of the endoplasmic reticulum is protein synthesis. Why the protein synthesis? The protein synthesis is because of the presence of ribosomes. Understood, student? The protein synthesis main function is because of the presence of ribosomes. If there were no ribosomes present on the surface of the endoplasmic reticulum, then absolutely there would be no protein synthesis function. This is the main function of the rough endoplasmic reticulum. Also, it also synthesizes general proteins and some molecules it can produce. Next, smooth endoplasmic reticulum. Then students, you may ask me one question. Okay sir, rough endoplasmic contains the ribosomes, therefore they are involved in the protein synthesis, a very much important function in this cell. Now the smooth endoplasmic reticulums, they have no connection with the ribosomes. Then what are these? Then what they will do in our body? Then what the function they are performing inside the cell? Okay. But students, let me tell you, the smooth endoplasmic reticulum, what happens if it cannot synthesize the protein? But it is allotted with the unique enzymes which are helpful, which are useful for the synthesis of various hormones in the body. Okay, what hormones the smooth endoplasmic reticulum will be synthesizing? There are a lot of functions that the smooth endoplasmic reticulum is synthesizing, or they are giving to the cell. The first is the production of hormone estrogen, progesterone. Okay, this is the main hormone in the human body, and also the androgen and testosterone. Okay, these are all the important biochemical substances, you can say hormones, which are needed in our body. So these are being produced by the smooth endoplasmic reticulum. So this much is not function of the smooth endoplasmic reticulum. It also have many major roles to perform in the body. Okay, like in liver. In the liver cells, the smooth endoplasmic reticulum which it is helping in the detoxify of hazardous substances such as alcohols and pesticides and carcinogens, etc., etc. These are eliminated by the smooth endoplasmic reticulum. And also in the liver, the smooth endoplasmic reticulum, the enzymes which are produced by smooth endoplasmic reticulum, the enzymes produced by smooth endoplasmic reticulum helps to release glucose into the bloodstream. Dear students, and the tail doesn't over here. And another important function of the smooth endoplasmic reticulum is it removes phosphate group from glucose 6 phosphate. In biochemistry you might have studied that the glucose 6 phosphate is present, and by the dephosphorylation of the glucose 6 phosphate, the pure glucose molecule is obtained. So this dephosphorylation is mainly done because by the help of smooth endoplasmic reticulum. Did you understand? Did you follow me? Next, very small Golgi apparatus. Golgi apparatus it is the sac of flattened membrane-bound sacs. It modifies, sorts, and packages proteins for secretion or delivery to the other cellular destinations. Example: a protein molecule is synthesized, and if a protein molecule is wrongly synthesized, it is a part of you only. Humans only make errors. If an error happens or the message changes, no, we don't want A, we don't want response of B, then there might be chances that by some changes, by modifying a little bit of protein A, it can be converted to response of B. That can be processed, that can be done with the help of Golgi apparatus. Is that clear, students? Is that clear? The main function of Golgi apparatus is it modifies, sorts, and packages proteins for secretion or delivery to the other cellular destinations. So that's all for today's class, students. We will meet in the next class. Thank you.